Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. Here with me today are my colleagues at NCCAT, Ed Eng. Joining from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt. And from the Pacific Northwest Cryo-EM Center, PNCC, is Omar Devolku. PNCC has invited today's speaker, Kenny Childers, who's joining us from Western Washington University. And Omar, will introduce him properly after a quick update from each of the 3 centers to keep you up to date on the resources we make available at no cost to the research community. For everyone joining us for the first time today, this webinar is a regular event. We host the last Thursday of the month, highlighting methods and tips and tricks that groups are using to collect and interpret the data they can obtain through the use of National Cryo-EM Center resources. So you can save the date now for our August talk. NCCAT will be hosting Elizabeth Stroop and Behrooz Efestani, who will be talking about the cryo-EM sample from Dante's Inferno and one solution to the air-water interface challenge. That sounds like a nice time for group commiseration. So if you're interested, you'll be able to register for that talk soon on the events page at cryoemcenters.org. And on that page, you can also find recordings from all of our past talks. So look out for registration links for upcoming talks. Back to today's talk. For Zoom logistics, please send questions at any time using the Q&A feature, not the chat, but the Q&A box. And Omar will be moderating questions at the end of the talk. And so let's go ahead and get started. So first, quick updates from the centers, and I will turn over first to Mike from S2C2. Go ahead, Mike.
Hi, I'm Mike Schmidt from the Stanford SLAC Cryo-EM Center. As for the other centers, we have 4 cryos, 2 other lower voltage microscopes. We use these for high resolution data collection, for training, and for specimen preparation help. Come and see us at slack.stanford.edu. And a special shout out to the people that are listening to us from the M&M conference in Minneapolis, taking their time out from attending the talks there to listen to our seminar today.
All right, thanks, Mike. I'll turn over to Ed next.
Hi, I'm Ed, coming from NCCAT. We're in the West Harlem Research Corridor. We offer 3 categories of access: data collection on our 4 Titan Crioses. They all have been recently upgraded with FFI to increase throughput. We offer screening that includes grid preparation and screening on our glacios, and training. We are all at the National Center program at Minneapolis. So if you're in our neck of the woods, come by. We have a booth all the way up until 2, and we're giving a raffle away for a chameleon. On another note, I did mention we're in the West Harlem Research Corridor. In October, we're offering an integrative structural biology symposium in conjunction with ASRC and NYSPC. Friday is the last date for abstracts. So if you would like to present a poster or present a talk, please do so. You can find more information on our website. Thank you.
Thanks, Ed. And I'll turn over to Omar.
Thanks, Christina. I am Omar Devolku, and I'm one of the microscopists at the Pacific Northwest Cryo-EM Center along the Willamette River in beautiful Portland, Oregon. We offer our users a seamless workflow from sample prep all the way to 3D reconstruction, including our screening access mechanism that allows for users to come on site and work with us directly through the entire workflow. PNCC has a number of microscopes, all equipped with K3s, Falcon 4i direct detectors, fringe-free imaging, and Gatan energy filters. We also offer in-person training for sample prep, grid screening, data collection, and processing. I'd encourage anyone with questions to ask either through the Q&A here or to visit our website.
All right, so that's everything from us. So Omar, I will let you introduce today's speaker.
Thank you again. It's really a joy to introduce Kenny, today's speaker, who's a postdoc in Clint Spiegel's lab at Western Washington University. Kenny holds a special place in PNCC's heart as he was our very first screening access user. Actually came out and worked with two microscopists here, Rosemary Haynes and myself, for a week, at the end of which multiple data sets were collected on samples that had never previously been frozen. Kenny's also gone on to attend a number of our training workshops and is now independent in virtually the entire cryo-EM workflow. So he's really a success story in PNCC's eyes. I'm really excited to hear him speak today about that and other work that has recently graced the cover of Blood. So Kenny, take it away.
Yeah. Thanks a lot, Omar. That was really really nice of you. All right, let's go ahead and get this. Then I'm just gonna hide this. Okay. Let me just get a laser here. Okay, right. So thank you all very much for attending to today's webinar. It's really a treat to be here. I've attended 2 of these so far in the past, so it's quite an experience to. It's a full circle experience to be here and actually presenting my own cryo-EM work. So in my talk today, I'm really going to go over sort of the story that Omar introduced at the beginning about how we started at PNCC and really the success that we've had through using their facilities at PNCC. Before I get started, I really quickly just want to emphasize that at Western Washington University, if you haven't heard of us, we primarily work with undergraduates and master's students. We don't have any facilities for cryo-EM. Before this, we've historically used crystallography in our lab. So really, it's the facilities and the personnel at PNCC that have allowed us, or that have been the driving force in the success of this project. So I'll go ahead and get started. So in our lab, we focus on the protein blood coagulation factor VIII, which is shown here in blue, and its involvement in the blood coagulation cascade. So in our bodies, factor VIII is secreted and it binds to this protein here called von Willebrand factor, which is shown in light blue, and it circulates in our bloodstream, which prevents premature proteolytic degradation of factor VIII. But in the event of a blood vessel rupture, thrombin will go and cleave factor VIII to generate activated factor VIII, which is depicted here as factor VIIIa. Thrombin will cleave factor VIII, it will dissociate from von Willebrand factor, and it will bind to these platelet lipid membranes and form a complex with activated factor IX to form what's termed the intrinsic tenase complex. And from here, this complex will bind to factor X to generate activated factor X, and that will ultimately lead to the formation of a fibrin clot to stop that bleeding from occurring. So really quickly, I'll go through what factor VIII sort of looks like. Factor VIII is a heterodimer. It's composed of a heavy chain and a light chain. Its overall molecular weight is about 180 kilodaltons or so. So this is a recent cryo-EM structure that came out a couple years ago. And this is a cryo-EM structure where they were able to bind factor VIII to a fragment of von Willebrand factor. And we were able to see how actually von Willebrand factor is binding. It actually binds predominantly to the C1 and the C2 domains shown here in orange and yellow. And remember, it's that binding of von Willebrand factor to factor VIII that's going to prevent premature proteolytic degradation of factor VIII. So it's really going to increase the circulatory half-life of factor VIII and allow it to, when there is a blood vessel rupture, factor VIII can be activated and we can generate that fibrin clot. The C domains are also going to be the ones that are going to bind to platelet membranes. They have these little solvent-exposed hydrophobic loops at the tips of them, and this is what's going to dock onto those platelet membranes. And then from there, it's going to form that complex with activated factor IX, shown here in pink. And those binding is, we think it's going to bind predominantly to the A2 and A3 domains of factor VIII, although we don't really know for certain because there is no structure of this complex. However, this pathway can be disrupted in patients with a disease called hemophilia A. So in hemophilia A cases, patients will have a mutation to factor VIII, and that's going to disrupt one part of this blood coagulation cascade in factor VIII. So we can have mutations that disrupt, for example, the binding of thrombin and its activation. There can be a mutation that disrupts binding to, factor VIII to phospholipid membranes, or binding to activated factor IX or factor X, or binding von Willebrand factor. There can be some point mutation to factor VIII that's going to disrupt the factor VIII biochemistry. But ultimately, all this is going to lead to a reduction in the formation of a fibrin clot and uncontrolled bleeding events in these patients. So regardless of wherever this mutation is, the treatment for hemophilia A typically is going to be regular injections of a recombinant or plasma-derived factor VIII to help increase their circulating levels of factor VIII and allow them to form a fibrin clot in the event of a blood vessel rupture. However, in these treatment cases, about 30% of these patients are going to elicit an immune response to that factor VIII that they're getting regular injections with. This is going to occur most frequently in patients with who have little to no circulating factor VIII due to whatever point mutation they're having or they have. And because of this immune response that they have, it's typically going to require increased frequency and dosages of that factor VIII injection, which can be both a physical burden on the patient as well as a financial burden for them as well. So how does this immune response exactly work? Well, predominantly these antibodies are going to bind to the A2, the C1, or the C2 domains. And depending on where that those antibodies bind, it's going to disrupt some part of the factor VIII biochemistry. So for example, anti-A2 domain inhibitors can disrupt binding to thrombin or activated factor IX or factor X. Anti-C2 domain antibodies, shown here in yellow, they could maybe disrupt thrombin binding or factor VIII binding to lipid membranes, or maybe von Willebrand factor interactions. But ultimately, these antibodies are going to bind to that recombinant factor VIII molecule that they're being injected with, and it's going to disrupt that treatment process. So the overall goal in our lab is to structurally characterize these complexes of factor VIII bound to these antibody inhibitors. And if we can characterize those interactions, we can hopefully provide structural evidence to generate or engineer a recombinant factor VIII molecule that's less immunogenic. So that's the overall goal in our lab, is to really understand how these antibodies are binding, where they're binding, and to provide data to re-engineer a factor VIII molecule that's less immunogenic. So to date, our lab has solved a number of crystal structures of factor VIII, or isolated domains of factor VIII bound to these antibody inhibitors. Historically, we've just focused on the C2 domain, shown here in yellow. We can express that C2 domain in E. coli. It expresses really well, and we can form these complexes with antibodies. And we get some really nice data on where these antibodies are binding. However, since then, we've also expanded to using a full-length mature factor VIII molecule, this one shown here in the bottom. This is a recent crystal structure that came out from our lab, where we used a full mature factor VIII molecule bound to an anti-C1 domain inhibitor, shown in pink. So this is really great. So historically, like I said, we've been using X-ray crystallography to study these complexes. And this has generated a lot of really useful information. We've improved our understanding of how immunogenic the C1 and C2 domains are, shown here in orange and yellow. We've also helped refine our understanding of how factor VIII binds to von Willebrand factor, as well as to lipid membranes. However, today, there are a few limitations to these studies. First off, there is no structure of factor VIII bound to an anti-A2 domain inhibitor. And secondly, most of these antibodies that I've been showing you of these structures, most of them have come from mouse models of hemophilia A. So how this works is we take a mouse model, we knock out its factor VIII gene, inject it with factor VIII, it generates antibodies, and then we take those antibodies and complex them with factor VIII to start to get an idea of where these antibodies are binding. But most of these antibodies are not coming from patients, they're coming from mice. And so the talk that I'm going to give today is going to show how we've overcome both of these hurdles using cryo-EM. So before I get into it, Omar sort of talked about it in the beginning, but I wanted to go over it a little more, sort of my trajectory through PNCC from start to today. So back in May 2021, in the midst of the pandemic, Clint, my PI, we started talking about incorporating cryo-EM into our research. So he told me about PNCC, and together we wrote a general access proposal in May 2021, which granted us 2 years of time on the at PNCC, where we could send samples and we could work with them and hopefully get a lot of great data. So we got it approved in May 2021. We were assigned Omar as our SPOC. It was really great to introduce ourselves and start working with him. We sent him some samples. They weren't allowing any on-site visitors at the time because of the pandemic, but we sent him a few samples and he threw them on some grids just to see what happens. And of course, they all aggregated. It was terrible. We sent him the samples. He sent us back a report, and he just said, "Yeah, it's terrible. It's not great right now." So around this time, we were scratching our heads figuring out what to do next. And then Omar emailed me saying that PNCC had just come out with a screening access proposal mechanism. And the purpose of this was to allow researchers who really didn't have a full grasp of their particle, of their protein, and how it would behave on grids. And maybe they don't have the means to study it at their own institution. So if we got a screening access proposal, it would allow us a week of time at PNCC where we could really screen our sample on different grids, and we could just screen it on a bunch of different conditions and figure out what works for this protein rather than waste our time in the general access proposal. So we wrote that new proposal and got it approved in March 2022. Around that time, I also attended a sample prep workshop in person at PNCC. This was right when they started opening their doors again. So this was me and my cohort, and we all came down there and got hands-on training on how to freeze and clip grids, which was really great because, again, at the institution I'm at, we don't have a VitraBot. We don't have access to this stuff. So PNCC kindly opened their doors and gave us hands-on training on how to actually operate a VitraBot and how to appropriately freeze and clip grids. Shortly after that, I attended my first on-site visit, which was through the screening access proposal. So I went down there, I brought a few of my samples, and I just spent a week training and further, freezing and clipping grids. And then I got to optimize a bunch of different conditions. One of the catalysts that really turns out that was really important for our protein was actually using grids that have a continuous substrate on them. So we were, in the first few days while I was screening with Omar and Rosemary, the protein was just constantly aggregating, as you see here in this image here. And Omar, during, I think it was the second or third day, he pulled me aside and he was like, well, we have these other grids that they have a continuous substrate, and they're really for finicky proteins that don't like to get in the holes, and they don't stay frozen in there into single particles. So let's go ahead and try them. And it turns out that was the key. That did everything. It brought our particle into the hole, and we were able to collect high resolution data on it, as well as we were also able to troubleshoot some protein concentrations and some additives as well. So that was last summer. I attended a second on-site visit back in February, where I also got training on the Arctica. And this was really important for us because not only did I get hands-on training at the institution, but I was able to get train on the Arctica to use it remotely from our home institution. So rather than having our SPOCs prepare our grids and screen them themselves, we know they're busy and they have a lot of other groups that they're assigned to, we could screen them ourselves. So just whenever the microscope's free, it allows me to hop on and screen them by myself. So that was really able to help increase the throughput of our project. After that, I attended a data processing workshop because up until this point, I'd really been bumbling around by myself, processing data and trying to figure out how things work. So Omar got me into this data processing workshop that I attended for a week remotely, and that helped train me on using Relion and CryoSpark, which again is provided through PNCC. We just recently got renewed for a general access proposal for another 2 years, which is really exciting. We're happy to continue this collaboration. And then just last week, I just got back from my third on-site visit at PNCC. So really, hopefully that gives you an idea of my trajectory through PNCC from start to finish. Again, about 2 years ago, I really had no idea I didn't have a thorough understanding of cryo-EM, and PNCC has really helped to evolve this understanding in this project. So in my talk today, I'm going to present 3 cryo-EM structures that have all come out of the PNCC through our collaboration. It's worth noting that the first 2 structures I'm going to present were the grids for these proteins were prepared during the screening access visit last summer. And the third structure was prepared on my second on-site visit this past winter. So, and remember, we're looking at factor VIII, and we're looking at understanding this antibody inhibitor interaction. So all three antibodies that I'm going to be presenting to you today, they all came from hemophilia A patients. And the first one, this antibody is termed NB33. Up until this point, researchers have shown that it blocks factor VIII binding to lipid membranes as well as von Willebrand factor, and it does this by binding to the C1 domain. It was originally reported about 20 years ago from a patient with mild hemophilia A. Some subsequent research found that you could just knock out that antibody binding by just knocking out 3 residues at the C1 domain, which are shown here. Those 3 residues are also shown to interact with von Willebrand factor from that recent cryo-EM structure that came out a couple of years ago. And then through some HGX-MS data that came out in the JBC paper, they also found that it binds not to 1, but to actually to 2 loops right here in the C1 domain. We think that's where that antibody is somewhere binding. So we were able to solve the cryo-EM structure of factor VIII bound to this antibody. We were able to solve it out to about 4.2 angstroms. We were able to confirm a number of those residues that previous researchers had hinted at, including this lysine. We have an arginine here, as well as a number of hydrophobic interactions of phenylalanine and an isoleucine here. So this was great. We were actually able to solve this was our first cryo-EM structure of factor VIII bound to a patient-derived antibody inhibitor. This was really exciting. And not only that, but we were able to confirm a number of previously reported interactions between factor VIII and this antibody inhibitor, which is shown here in purple. Looking at the electrostatics of this interaction, interestingly, actually this antibody uses a negatively charged acidic cleft to target these positively charged residues on the C1 domain. So it has these negatively charged residues, and it's going to target these 2 lysines and 2 arginine residues, and it's going to dock on there. And that's really how it's going to specifically target that region of factor VIII. Now, one other interaction with factor VIII that I haven't mentioned yet is actually the clearance of factor VIII. So factor VIII is circulating in our bloodstream. It's preventing those bleeding events from occurring. But eventually, we need to get rid of factor VIII. We need to clear it out of our bloodstream. How does it do that? Well, there are cells that use this transmembrane protein called LRP1. LRP1 is going to bind to regions of factor VIII, and that cell is then going to perform endocytosis and sequester it, and subsequently use that for processing and antigen presentation in dendritic cells. Some HGX-MS data came out, and they looked at this LRP1 protein is actually going to target a number of positively charged lysine residues that are scattered across the factor VIII molecule. So shown here in red are those those lysine residues on factor VIII that researchers believe are going to be targeted by LRP1. It's going to bind to those lysine residues, and it's going to perform endocytosis on factor VIII and remove it from our bloodstream. And it turns out that with NB33, when we bind factor VIII to NB33, it actually blocks binding of LRP1, and it's going to allow factor VIII to continue to circulate in our bloodstream. So by binding NB33 to factor VIII, it increases the circulatory half-life of factor VIII. It's kind of interesting to think about. So now we can look at this structure that we just solved, and it actually turns out this antibody uses negatively charged residues to target those positively charged residues on the C1 domain. And so the last thing I'll talk about with this structure is there's one previous structure that came out from Clint back in his grad program, where he actually solved the structure of the isolated C2 domain, shown here in yellow, bound to one patient-derived antibody termed BO2C11. And this antibody actually binds similarly to the NB33 antibody that we just recently solved. BO2C11 also binds to several positively charged residues, these 2 arginines shown here. And actually, there has been research that shows that when you bind BO2C11 to factor VIII, it's also going to inhibit the clearance of factor VIII. So we have these 2 different antibodies that bind to different regions of factor VIII, but when you bind them to factor VIII, they both bind to positively charged residues, and they also inhibit the clearance of factor VIII as well. However, if we take just factor VIII and we knock out these positively charged residues, it turns out they have different results. So if we take the positively charged residues in the C1 domain and knock them out, we get a statistically significant drop in the immunogenicity of factor VIII. So not only are we looking at the clearance, but we're looking at how do those positively charged residues influence the immune response to factor VIII. So if we take those positively charged residues in C1 domain and knock them out, we can reduce the immunogenicity of factor VIII. But that's not the case with the C2 domain. Previous research has shown that when we knock out the residues in the C2 domain, there is no difference in the immune response. So it's just with the C1 domain. Apparently, what we're seeing so far is that when we knock out those arginines and lysine residues, we can reduce the immune response to factor VIII. So it's intriguing to think about these two different antibodies that bind to similar places with a similar mechanism, but these residues themselves have different roles in the immune response to factor VIII. So all of this was recently reported, as Omar just mentioned, in the journal Blood. We just got the cover of this. I think came out a couple weeks ago. So we were really happy to get this out there. We had Spock's Omar and Rosemarie on there. It was just really great to put this story together and have it out there. So we were really pleased with this report. Okay, so I'll go on to the next structure. So this next antibody that we ended up characterizing is an anti-A2 domain inhibitor. This is an antibody called NB11B2. It was originally reported in a patent about 15 years ago or so. And researchers there found that it binds somewhere to the residues 484 to 509, which I'm highlighting here in the A2 domain. So we think they thought somewhere this antibody is binding here to the A2 domain. Our collaborators took this antibody and they found that this antibody will disrupt, it will disrupt formation of the intrinsic tenase complex. However, we also have some research that show that when this antibody binds, it's not going to disrupt thrombin cleavage. So remember, in the biology of factor VIII and the life cycle of it, when thrombin cleaves, it's going to cleave a linker between the A1 and A2 domains. So when you have it go from the absence to the presence of thrombin, you can see how the A1 and A2 domains separate. However, in the case of this antibody, it doesn't appear to disrupt that thrombin cleavage. And lastly, I'll just go over the general antibody response to the A2 domain. So what you're looking at here in this graph is from a paper that came out in the 90s. We're looking at 5 different patients and their immune response to the A2 domain. And on the x-axis, these are a bunch of different mutations that these researchers made to the A2 domain. They wanted to see what happens if we mutate this residue, how do the antibodies bind and inhibit factor VIII? And really the takeaway message from this graph is it's not black and white. There are these antibodies that bind to factor VIII into the A2 domain really will bind to discrete places and discrete mechanisms. So just because one antibody binds to this location doesn't mean that all anti-A2 domain inhibitors are going to bind to that same place and the same mechanism. They all bind very differently. And the last thing I'll just mention about this anti-A2 domain response and studying it, the A2 domain historically has just been very difficult to work with. As you can see here in this, the structure of factor VIII, the A2 domain actually packs in very closely to the A1 and A3 domains. So it has a lot of these buried hydrophobic regions. So trying to recombinantly express just the isolated A2 domain is quite challenging. It's a lofty goal. So that can be could be one of the reasons why there has just been a lack of structural information on the immune response to the A2 domain. So we took this sample down there. We complexed the NB11B2 with factor VIII, and we were able to get a decent a data set from this protein complex. What you're looking at here is a locally refined map of the structure down to about 3.8 angstroms. We were able to dock this antibody in and model it. And it turns out we were able to further confirm where this antibody is binding and refine those residues that are interacting. Predominantly, while the previous structure utilized an electrostatic interaction, here we're looking at more hydrophobic interaction. So you can see here on the top and bottom images, there's a bunch of these hydrophobic residues that this antibody is going to use to bind to factor VIII and sequester those solvent-exposed regions. And you can see that further in the next slide. Oh, one more slide. when we actually look at this antibody binding, it also doesn't seem to disrupt a putative where we think the factor IX molecule is binding to factor VIII. So rather than directly inhibiting and competing with factor IX for binding to factor VIII, what we think is happening is it's going to bind to factor VIII and use some sort of allosteric mechanism to disrupt the activation of factor X by forming the intrinsic tenase complex. So somehow, we don't really have a thorough understanding of this yet. Somehow this antibody is going to bind to a region of factor VIII that isn't where factor IX is binding, and somehow, possibly through allostery or maybe disrupting the factor X binding site, it's going to inhibit the intrinsic tenase complex activity. So that's sort of our idea, the mechanism, how this antibody is working. We don't really have a thorough understanding of that quite yet. And again, this is just looking at the electrostatics of these antibodies. While the first two antibodies that I introduced in the previous part, they use these negatively charged paratopes to bind to factor VIII. NB11B2 is actually going to use this hydrophobic sort of a loop that shoots out and is going to bind to those hydrophobic residues on factor VIII. So the last thing I really quick want to talk about with this structure before I move on. So in all these structures of factor VIII that our lab has characterized to date, one thing that I haven't talked about is the conformational change that the C2 domain can adopt. We've actually characterized a number of different rotational and swing movements that the C2 domain can adopt relative to the rest of the factor VIII molecule. So in two of the structures that previous students have characterized, the C2 domain adopts more of a swinging or a rotating structure. So it kind of rotates around, and you can see these arrows, and it kind of gives you an idea of how it's twisting. However, in a third structure, one of our grad students found that the domain doesn't twist, but the C2 domain actually swings out and adopts about a 20-degree swing relative to the rest of factor VIII. So if we look at the cryo-EM structure of the NB11B2 bound to factor VIII, we actually found that this C2 domain actually does adopt a 20-degree swing. So it actually supports this bottom structure, this bottom conformational change that we've seen in a previous crystal structure. So we were able to see this by expanding our refinement. We went to a general non-uniform refinement, and we brought it up to about 4 angstroms. And we can see that when we do that, we see that the C2 domain actually swings out from the rest of the factor VIII molecule. The biological implications for this, we haven't really fully understood quite yet. Why would factor VIII want to have this C2 domain that's swinging out? And how would there be this allosteric network connecting from the A2 domain down to the C2 domain to provide this C2, to induce this C2 domain movement? We haven't quite fully understood that yet, but it's something we're seeing in our cryo-EM map. And it's something that we can actually see in cryo-EM versus in crystallography, which can be quite challenging when you have a domain that's swinging around a lot. Okay. And in the last structure I'm going to go over today, or the last antibody, this is an antibody termed NB2E9. This antibody binds to the C1 domain, and it disrupts von Willebrand factor from binding. It was first reported about 20 years ago. Researchers found that this antibody actually binds near this region where the patient has a missense mutation. It's arginine 2150 to histidine. That's where this little star is shown. And actually, this antibody doesn't disrupt that variant. It's only binding to wild-type factor VIII. But if you give this, if the patient is given its own missense mutated factor VIII, this antibody is not going to bind to that region. Furthermore, that variant is also going to disrupt von Willebrand factor from binding too, which is shown, which we're looking at here. So this is sort of the side angle of the C1 domain. This is where we think the acidic part of von Willebrand factor is going to bind to the C1 domain, and that's sort of how those electrostatics are going to mediate that binding. Some binding studies that came out back in 2017, they found that one single point mutation, it's a glutamate to an aspartic acid at 2066, that can knock out all NB2E9 binding. And one other really interesting thing about this antibody actually has a glycosylation group at the paratope. This actually only occurs, it's thought to occur in about 20% of naturally occurring antibodies. So this glycosylation group occurs at one of the CDR loops. And actually, when we deglycosylate that antibody, as we're looking at here, if we deglycosylate the antibody, the inhibitory activity of that antibody drops by about 50%. But the affinity for the antibody to factor VIII remains the same. So really, it's that glycan on the antibody that is dominating how it's inhibiting factor VIII somehow. And because of actually that decrease in inhibitory activity with this antibody, researchers have also begun to explore how it can be used as an antithrombotic, an antibody that can bind to factor VIII and actually reduce the thrombotic activity of it and be used for patients who are clotting too much. So we're not looking at hemophilia anymore. We're actually looking at people who have diseases like thrombosis. So we saw this cryo-EM structure. We actually got it even better than the previous two structures I reported on earlier. This actually came out to about 3.4 angstroms or so. And we were able to characterize a number of interactions, including that glutamate to aspartic mutation. Remember, that's a mutation that abolishes all binding to this antibody. So it turns out that glutamic acid is binding to a number of hydrophilic residues shown here in green. As well as if we kind of turn around on the backside, we can see a number of hydrophobic interactions shown here with phenylalanine and a couple of tryptophan residues as well. Actually, due to the quality of this map, we were actually able to even model in some known glycosylation sites, both on factor VIII, which are shown here in asparaginase 280 and 1810, as well as that glycan group I previously mentioned. Remember, this is the glycan that if we deglycosylate it, the inhibitory activity of this antibody drops by about 50%. So we can actually see in our cryo-EM map, we can see where that glycan is coming out. And interestingly, that glycan, if we look at our map, it's not actually making direct contacts with factor VIII. Remember, I mentioned earlier that if we deglycosylate this antibody, the affinity for factor VIII remains the same. So we can see in our cryo-EM map that this glycan is not binding directly to factor VIII, but instead, what it appears to be happening is it's going to disrupt von Willebrand factor from binding to factor VIII. So if we take the cryo-EM structure of factor VIII bound to von Willebrand factor and we overlay it with our structure shown here, we can see steric clashing between the glycan and von Willebrand factor shown here in pink. So the mechanism of action, we think with this antibody, is that glycan is going to sterically block von Willebrand factor from binding to factor VIII, and it's going to increase the clearance rate of factor VIII. And so the last thing I'll go over with this structure is sort of comparison between a previous structure that we found using a mouse-derived antibody 2A9 and this structure NB2E9. So previous structure came out from two really talented crystallographers in our lab, Joey Gish and Lexi Jarvis. They determined a crystal structure of factor VIII bound to this antibody 2A9. This antibody binds both to the C1 and the A3 domain, shown here in cyan. And if we overlay the two structures between NB2E9 and 2A9, we can see there's actually a shared epitope between both of these antibodies. And this is interesting to think about because we're not only looking at a human response to factor VIII, but we can compare them to the immune response people typically see in mouse hemophilia A models, which are typically used in a lot of research labs. So we look at the immune response between 2A9 and NB2E9, we see a shared epitope between both of these antibodies shown here in purple. And we also see differences too. The 2A9 actually makes partial interactions with the A3 domain. Meanwhile, the NB2E9 kind of tilts downward and it makes more interactions with this region on the C1 domain shown here in red. But again, we're seeing this overall shared response between mice and humans toward factor VIII right here on the C1 domain. And this is the first structural evidence that we've seen for this immune response. So hopefully what I've shown you today is really the benefit of cryo-EM for our research and how it's really been a catalyst toward furthering our understanding of the immune response to factor VIII. We were able to look at the electrostatics between one antibody, NB33, which uses this negatively charged cleft to target positively charged residues on the C1 domain. We were able to report on the first structure of factor VIII bound to an A2 domain inhibitor. And we were also able to further support this conformational rearrangement to the C2 domain. And lastly, we were able to characterize this anti-C1 domain inhibitor that uses a really novel glycan group. And due to the high quality of the map, we were able to model that in, as well as a number of other glycosylation sites on factor VIII. So that's really been the benefit of cryo-EM and how it's really helped our lab. And so I'll just close by thanking you all for your attention. This has been a real treat. I'd like to thank my lab. I always like to tell people it's just astounding that I work with these really young researchers. I tell people, I work this demographic of 20 to 30 somethings, and they just have this intrinsic interest in research that hasn't been squashed yet by grad school. Historically, I just work with undergraduates, and they have just a genuine interest in biochemistry, and it's an absolute treat and an honor to work with them. And then I really want to thank collaborators, especially Omar and Rosemarie. It's been an absolute joy working with them and getting all their knowledge and dealing with me and my fumbles. And when I go down there, it's just an absolute treat going down there and working with them. And then lastly, I'll thank funding sources, the NIH and the NHF. And with that, I'll take any questions you may have. Thank you. Go ahead and stop sharing.
Awesome. Thank you, Kenny. That was marvelous. And I think I've got a couple questions as well, but I see some in the Q&A, so I'll let Omar send you some questions from our attendees first.
Thanks, Christina. I'll second the notion that was a really nice talk. The audience was almost stunned into silence, but the questions did come through. So this question came in before you went on to explain, but maybe I can give you the chance to revisit or add any information you'd like. If the glycans are removed by PNGAs, do they still prove to be effective or even more effective? The antibody.
If the glycan is removed by the antibody, the inhibitory activity drops by about 50% or so. So it's dominating the inhibitory activity of the antibody, but it's not entirely. It still has some inhibitory activity. It just drops by about 50% or so.
Okay. Can you describe your method for obtaining and purifying patient antibodies?
A short answer, no, I can't. We get them from our collaborators. They ship it to us, and we do. What I can tell you is they ship us the IgG antibodies, and we just cleave them with papain or FVIII, mostly papain, and we just purify the Fabs from there. It's super quick and easy. Write to me if you would like the paper for how they recombinantly express those antibodies, those patient-derived antibodies. It's a very important work that they do, and that's really been a catalyst for us.
So I'll take this chance to ask a question before letting Christina ask hers. You mentioned that about 30% of patients can't tolerate factor VIII therapeutic directly because of an immune response. Yet you show antibodies that inhibit the uptake of factor VIII from the bloodstream, let's say.
Right.
So MABs are a very popular therapeutic now. Is it possible that potentially an anti-LRP MAB to extend the lifespan of factor VIII in the blood? Or does factor VIII remain active itself with NB33 bound?
Right. It's sort of like it's an interesting thought, isn't it? It's like a chicken and egg type of thing. Right. So absolutely, that's one genuine interest, is to use a MAB or some maybe a short chain FV domain where it can bind to that portion of factor VIII and allow it to increase the circulatory half-life. But I will also say that that antibody is also going to block that region of factor VIII that binds to the platelet membranes. So it's not going to allow the clotting it's going to inhibit or disrupt the clotting process. That being said, there are also a number of other non-LRP1 proteins that mediate factor VIII clearance. So it's not the only one. There are a number of other hurdles. So factor VIII does bind to a lot of different proteins, and that's just one of them. So, yeah.
Thank you.
Mm-hmm.
Yeah, I'll pop in with a question real quick, and do feel free to send questions into the Q&A if you've got them. So actually, one just came up when you were answering the previous question. So I sort of missed that you were generating Fabs And I'm wondering if you've tried using full-length antibodies.
In my head, I have. I've definitely done that thought experiment before. No, we haven't. That would be really cool, wouldn't it, to sort of complex two at the same time? And yeah, no, we haven't. Definitely thought about it though.
Okay. Then I also had a question. So I know, so you're using a continuous carbon support to avoid aggregation, but often you need much lower protein concentrations as well. So I'm curious what concentrations you're working with, and if it is sort of lower than typical, since you have that support film.
I guess I wouldn't really know what's typical. What I will say is what the protein concentration we work with is about 0.1 mg per mil. We just do this large dilution of our stock protein. We complex it, dilute it in some heaps buffer, and get it down to about 0.1 mg per mil based off the A280 absorbance, and go from there. I will say, yes, that just on my last visit, actually, with Omar, one of our proteins that we also diluted down to about 0.1, it was quite crowded. They're very, very crowded on that carbon support. So I'm actually in the middle of processing it right now, and it is pretty crowded. So yes, there could be something to be said about using a lower concentration than normal with these particular grids.
And I'll chime in with just a little bit. I think we did a very quick calculation, and that corresponded to low single-digit micromolar concentrations.
Yes.
So we just had one question come in. Did you worry about the increased background when you were using the continuous carbon layer?
Yeah. It's definitely something that it is a concern for us, especially during the downstream data processing and picking particles. It can be a little challenging. So yes, it's something that's always in the forefront. We're always thinking about it, but you know what? If it works, it works. We are able to get decent data out of it. We're not after sub 2 angstrom data sets here. Factor 8, you're never going to get it past, let's see, right now the highest resolution is 2.9. So you're just never going to get it super high resolution anyways. So to answer the questions that we're interested in with this epitope mapping, it works.
And your alternative was an aggregated mess, right? So
yeah, that's it.
Another question just came in. Why not develop and use peptide mimetics of the antibody binding region to prevent antibodies from binding to recombinant factor 8 in patients?
Totally. That's a great idea. Yeah. And I think that's one of the thoughts, is that once we characterize where specifically these antibodies are binding, we can maybe generate sort of like a molecule that acts as a sponge for that immune response, where the patient can get this in a small little antibody, and rather than bind to the full mature protein, it can just bind to this little portion and allow factor 8 to perform its function. So that's definitely a good thought, and that's where we're going.
This might be a more an immunology question, but if you've given the recombinant factor 8 with the mutations which you found which had the binding site, what prevents the patient from recognizing that as foreign and generating antibodies to the mutated factor 8, which stopped one antibody?
Definitely. Right. And that's not to mention patients don't have a monoclonal response. They have a polyclonal response.
Oh, sure.
There are lots of antibodies that bind. So this is just one piece of the puzzle. You're absolutely right that if we make a mutation, who's to say whether or not that's actually going to disrupt an antibody from binding and inhibiting factor 8? I don't have an answer for that. That's a really good point. And that's something that will eventually lead to, but first things first is to get an idea of where these antibodies are binding. But you're totally right.
Well, that looks like it might be all of the questions. I'll take this moment to thank you again, Kenny. It's really been great working with you, and this was a great talk.
Thanks.
Yeah, and actually, I'll chime in with one more to give everyone who needs a second to type, if they've got one last question, to type it in. Last call for questions. But so since you're using then sort of the same antigen for all of these, now that you've sort of got conditions pinned down, how easy is it to make to sort of prepare grids using a new fab? Like if you,
Right.
do you are you starting from square one optimizing every time, or do you have a robust system now since your antigen at least is the same?
Right. That's great. So in terms of actually studying those antibody and factor VIII complexes, I think you're right. I think we have come up with a pretty robust system for for isolating these samples. However, I will say that's not the only structure that we're interested in. We're also interested in other portions of factor VIII biochemistry that are not related to its immunology. So, for example, other proteins that bind to it, like von Willebrand factor and factor IX, and those require more optimization and screening. So, for example, just last week when I was down with Omar, we found out that CHAPS worked great. CHAPS is awesome, and I never had any CHAPS in the antibody complexes. But having a tiny amount of detergent seemed to really do the trick.
That's cool. Another good tip. Thank you.
Yeah.
Looks like we got a question about how someone can join the mailing list for these webinars.
Yeah, so actually, everyone who's attending will send out an email once we have the recording posted, and there will be information about that. So if you're here now, expect an email from us following up, and we'll get you that information.
And our normal outlet for advertising these is the CCP EM and all the different EM mailing lists. And if you're on those mailing lists, you'll probably get a number of invitations from all the centers for all these seminars. We work for our mailing list. We work through our old users. We work any other information you can give us about how to contact people that haven't heard of us yet. That any kind of other sources you use for information about these seminars, we can add that and invite everyone. And you mentioned, Kenny, you mentioned that you've attended these before. How did you hear about us?
I think I got an email. You know what? I think my Clint, my PI, is on Twitter, and so he told me about one of the webinars was using nanodiscs, and I just started using them, so I hopped on and listened to one of them. Yeah.
Okay, well,
that's it.
All the sources of dissemination we can think of. That's
right.
Okay, thanks.
Yep.
So last minute question, Kenny. Do you want to take a moment to discuss why you use CHAPS?
Because we used CHAPS last week when we were observing some signs of aggregation. And this was for a non-antibody complex. I should specify this. We were viewing some during from day 1 or day 2 last week. And so I had brought along some CHAPS, and so I just threw it in there, and that really made a world of difference. So, yeah.
And was it sort of sub-CMC concentrations of CHAPS or closer to CMC?
I think it was closer to CMC, but it was night and day on when we were screening on the Arctica. Yeah.
Great tips.
Okay. Definitely.
You're getting a lot of thank yous in the Q&A.
Great. Yeah, that's awesome.
All right, so I'm going to go ahead and